Look at your hands. If you're over 60, you have probably noticed something that was not true at 30. They look like your mother's hands, or your father's. The shape of the knuckles, the way the tendons stand out, the particular pattern of veins on the back. 20 years ago, you might not have seen the resemblance. Now it is unmistakable. You are not imagining this. There is a specific physical reason it is happening now and not earlier, and it changes what you think DNA actually does. 2 complete sets of genetic instructions live inside you, one from your mother, one from your father, roughly 3.2 billion base pairs from each, zipped together into 46 chromosomes in the nucleus of nearly every cell in your body. But those instructions are not a blueprint. A blueprint is a picture of the final product, an architect's drawing that shows what the building will look like. DNA contains nothing like that. There is no picture of your face in your genome, no diagram of your hands, no sketch of the person you will become at 70. If a geneticist sequenced your entire genome the day you were born, they could not have drawn your face from the sequence. They could not have predicted the exact shape of your hands, the precise curve of your nose, the specific way your father's jawline would emerge from beneath your skin 6 decades later. The instructions are there. The picture is not. The picture only emerges when the instructions run, and what they produce depends on everything that happens while they are running. DNA is source code. It contains instructions for building proteins, molecular machines that interact with each other and with the environment to produce outcomes not written anywhere in the genome itself. The same DNA sequence, running in different conditions, produces different physical results. This is not theoretical. It is happening inside you right now. The skin cells on the back of your hand and the nerve cells in your brain carry identical DNA, the same 3.2 billion base pairs. But the skin cell produces keratin and melanin. The nerve cell produces neurotransmitters and ion channels. Same code, different runtime environment, different output, in the same body, at the same time. Identical twins demonstrate this at the scale of a whole person. Twins who share 100% of their DNA at conception diverge physically over a lifetime. By middle age, their methylation patterns, the chemical tags that control which genes are accessible, differ at thousands of gene sites. A phenomenon called epigenetic drift, driven by decades of different diets, different stress levels, different microbial ecosystems in their guts. Their faces diverge. Their disease risks split. One twin may develop type 2 diabetes while the other does not, despite identical genomes. Even their fingerprints, formed in the womb, are different from birth, because the fluid dynamics of amniotic flow across the developing fingertip pads create unique friction ridge patterns through mechanical buckling, sensitive to the exact position of each fetal hand. Same code, different runtime, different output at every scale. What you learned in school that inheritance works in simple ratios. One gene for eye color, brown is dominant, blue is recessive. One gene for height. Gregor Mendel, breeding pea plants in the 1860s, showed that traits pass from parent to offspring in clean, predictable patterns. And for peas, that is true. Flower color, seed shape, each controlled by a single gene. The mathematics was beautiful, but look at your face. It is not controlled by one gene. Genome-wide association studies, scanning the DNA of hundreds of thousands of people, have identified over 12,000 genetic variants that each contribute a tiny amount to adult height alone. Eye color involves at least 16 genes. The shape of your nose, the structure of your jaw, the way fat sits beneath the skin of your cheeks, hundreds of genetic variants, each nudging development by a fraction of a millimeter, collectively producing the face you see in the mirror. No single gene gave you your father's nose. Hundreds of genes, each contributing a barely measurable amount, assembled during your development into something that resembles both parents without replicating either. You are not a blend. You are a mosaic. Millions of small genetic influences, inherited from 2 people, combined into something neither of them quite predicted. Now, here is something most people never learn about the moment they came to exist. The genetic material you received from each parent was not a clean copy of either of their chromosomes. It was shuffled. When your mother's body produced the egg cell that became you, one specific cell, in one specific month, In one specific year of her life, her chromosomes did something extraordinary. The matching pairs, one she inherited from her mother, one from her father, lined up next to each other inside the dividing cell. And then the cell deliberately broke them. An enzyme cut the DNA backbone at multiple points, and the broken ends swapped. Segments from your maternal grandmother's chromosome joined to segments from your maternal grandfather's, creating a patchwork that had never existed before. Your father's sperm cell went through the same process, shuffling his parents' chromosomes at different random breakpoints. Breakpoints were essentially random. The egg that became you carried a combination of your maternal grandparents' DNA that your mother's body had never produced before and would never produce again. The sperm that became you carried a combination of your paternal grandparents' DNA equally unique. Mathematically, the number of possible combinations from any two parents, considering just the independent sorting of 23 chromosome pairs before even counting the crossover shuffling, is roughly 70 trillion. The probability that your parents could produce another child with your exact genetic combination is approximately 1 in 70 trillion. You are a one-time event. Your siblings share many of your genes, but the specific patchwork, which segment from which grandparent, stitched at which breakpoint, is yours alone. If your parents had conceived a child 1 month earlier or 1 month later, a different egg and a different sperm would have met, carrying different patchwork shuffled at different breakpoints. That child would have been your sibling. It would not have been you. The specific combination of genetic variants that built your face, your metabolism, your predisposition to particular diseases, your particular way of processing the world, all of it determined by molecular breakpoints that formed during one specific cell division in your mother's ovary and one specific cell division in your father's testes in one specific month decades ago. That is 1 in 70 trillion, and that number does not account for the next layer. When the egg and sperm that became you were produced, the DNA copying machinery made mistakes, roughly 60 to 70 of them, tiny errors in the 3.2 billion base pairs being replicated, missed by the proofreading enzymes that catch most copying errors. These are called de novo mutations, and neither of your parents carries them. They are new. They appeared for the first time in you. Most of your 60-odd novel mutations sit in non-coding regions and do nothing detectable. A few may fall in regulatory sequences and subtly change when or where a gene activates. Rarely, one alters a protein. You will likely never know which of your traits, if any, trace to these new variants. But they are there. 60 genetic sequences that exist in your body and nowhere else on Earth. Not in your parents, not in your siblings, not in any other human being alive or dead. If you have children, some of those 60 variants were passed on, shuffled into the next generation's patchwork, carried forward. If you do not, they end with you. 60 sequences that appeared for the first time when the egg and sperm that became you were produced existed for exactly one human lifespan and will never appear again. Every person who has ever lived carried their own set. Every person who has ever died took some with them. You are not a copy of your parents. You are a 1 in 70 trillion recombination plus 60 novel mutations running in an environment neither of your parents experienced. The biological word for this is unique, not as flattery, but as a physical fact about the information content of your genome. But the genome you inherited is not the whole story. There is a second layer of information sitting on top of your DNA, chemical tags that control which genes each cell can read. And these tags are not fixed at conception. They can be altered by experience, by what happened to you, and, remarkably, by what happened to your parents before you were conceived. Your DNA is wrapped around proteins, and both the DNA and the proteins are decorated with these chemical tags. Methyl groups on the DNA itself, acetyl groups on the proteins. These tags act as locks and keys. A methyl group sitting on a gene's control region blocks the cell's reading machinery from reaching that gene. The gene is silenced, not deleted, not damaged, but physically inaccessible. Remove the tag and the gene becomes readable again. Different cells carry different patterns of these tags, which is why a neuron reads neuron genes and a liver cell reads liver genes from the same underlying code. This system is called epigenetics, meaning above genetics. And here is where it becomes personal in a way you may not expect. In the winter of 1944, the German occupation of the Netherlands imposed a food embargo that cut caloric intake to as low as 400 calories per day for millions of people. The Dutch hunger winter lasted roughly 6 months. Pregnant women carried their children through severe famine, some in the first trimester, some in the second, some in the third. The timing mattered. Decades later, Bas Heijmans and colleagues at Leiden University, publishing in Nature Genetics in 2008, examined the now adult children of those pregnancies, people in their 60s who had been in utero during the famine. They found that individuals exposed during early gestation had measurably different methylation patterns on the IGF-2 gene, insulin-like growth factor 2, a gene that regulates fetal growth and adult metabolism. The promoter region of IGF-2 was less methylated in the famine-exposed group than in their siblings conceived before or after the famine, meaning the gene was more accessible, more active for 6 decades. The consequence: altered metabolic programming. The famine-exposed group had higher rates of obesity, cardiovascular disease, and metabolic syndrome in adulthood. Their bodies had been programmed in the womb by maternal starvation to extract and store calories more aggressively, an epigenetic adaptation to an environment of scarcity that became a liability in an environment of plenty. 60 years later, those methylation differences were still detectable. The famine had not changed these people's DNA sequence, not a single base pair was altered. It changed which of their genes were accessible, and the change persisted for a lifetime. Think about what that means for you. Your parents' experiences, not just their genes, but what happened to their bodies before you were conceived, partially shaped the epigenetic landscape you were born with. Stress alters cortisol-related methylation patterns. Toxin exposure modifies detoxification gene expression. Diet changes metabolic gene accessibility. Michael Meany at McGill University demonstrated this with extraordinary precision in rats. Pups raised by attentive, high-licking mothers had lower methylation at the glucocorticoid receptor gene in the hippocampus, making the receptor more accessible, producing more receptors, resulting in a calmer stress response throughout life. Pups raised by inattentive mothers had higher methylation at the same gene, fewer receptors, stronger stress response, more anxiety. The difference was not genetic, it was epigenetic, produced by maternal behavior in the first week of life and stable for the animal's entire lifespan. And when the high-anxiety pups were cross-fostered to attentive mothers, the methylation pattern partially reversed. Behavior changed chemistry. Chemistry changed gene accessibility. Gene accessibility changed the animal. What this means for you is direct. The quality of care you received as an infant, the specific pattern of contact, feeding, stress, and safety in your first years of life may have altered the methylation patterns on your own stress response genes, not your sequence. Your accessibility, the way your body handles cortisol, the speed of your stress recovery, the baseline anxiety level you carry. Some portion of these traits may trace not to the genes you inherited, but to the epigenetic marks deposited by experiences you cannot remember, in a body too young to form conscious memories. You inherited your parents' genes. You also inherited echoes of their experiences, written in chemical tags that you carry in your cells right now, and your children may carry echoes of yours. Now, why do you look more like your parents at 60 than you did at 30? Look at the tendons on the back of your hand. At 30, they were hidden beneath a layer of subcutaneous fat. Now they stand out, and they stand out in the same pattern your mothers did or your fathers. The genes that built those tendons were always there. So why are you only seeing them now? The answer is not that you changed. The answer is that your genome has lines of code with delayed execution. Genes that were always there, always present in every cell, but were not strongly expressed until middle or late adulthood. The activation schedule is itself genetically encoded, promoter sequences and enhancer regions that respond to age-related changes in hormone levels, telomere length, and accumulated epigenetic modifications. Sean Carroll an evolutionary biologist at the University of Wisconsin-Madison, explained why this is possible in his book, Endless Forms Most Beautiful. The 5 fingers on your hand were patterned during your embryonic development by a signaling molecule called Sonic Hedgehog, named, improbably, after the video game character. That same molecule patterns the fins of a fish. The vertebrae of your spine were laid out by Hox genes, the same Hox genes that pattern the body segments of a fruit fly. Your eyes were built using a gene called PAX6, the same gene that builds a fly's compound eye, repurposed for a completely different optical design. What makes you human is not human genes. It is the timing, location, and intensity of gene activation, the switches, not the code. Carroll was describing differences between species, but the same principle operates across a single lifetime, within your body, right now. Some of your gene switches were set to activate decades after birth. This is the answer to the question you asked when you looked at your hands. Specific genes controlling how fat is distributed beneath your skin are expressed more strongly after 50 than before. The subcutaneous fat layer in your hands, the layer that kept the tendons hidden and the veins invisible at 30, thins as these genes shift the fat distribution pattern from peripheral to central. The tendons emerge, the veins become visible. The shape of the knuckles, which was always determined by your skeletal genetics, becomes visible through the thinning skin for the first time. Your mother's hands were not hiding inside yours. The genes that would reveal the resemblance were waiting for the right hormonal environment to activate. The MC1R gene, which influences skin pigmentation and hair color, has variants whose effects on skin texture and aging intensify after 50. IRF4 affects the rate at which collagen cross-links break down in your skin, with expression that increases in later decades, accelerating the specific pattern of wrinkling that you inherited from one parent or the other. The particular way your forehead creases, the specific depth of the lines around your mouth, the exact pattern of skin laxity along your jawline. These are not generic aging. They are your aging, running on your specific genetic variants, on a schedule that was encoded before you were born, your grandmother's cheekbones appearing in your face at 65, your father's hands emerging from yours. These are specific proteins, produced by specific genes, activated on a developmental timer that was set at conception, reshaping your tissue in patterns that echo the same genes running in your parents' bodies at the same age. I find this remarkable. The resemblance you noticed, the one that startled you because it was not there a decade ago, is not you aging into your parents. It is your genome executing code it has carried since conception, waiting for this moment to run. There is one more layer worth knowing. Every woman is a genetic mosaic, a discovery made by Mary Lyon in 1961. In each cell of a female body, one of the 2 X chromosomes is randomly silenced early in embryonic development. Which X is silenced, the mother's or the father's, is random per cell and permanent. The result, a woman's body is a patchwork of 2 genetically distinct cell populations. In calico cats, this produces the visible patchy coat. Orange patches express one parent's X, black patches express the other's. In women, the mosaicism is present in every tissue, but usually invisible. And there is one more fact that connects a mother to her children in a way that DNA alone does not account for. During pregnancy, fetal cells cross the placenta and enter the mother's bloodstream. Some of those cells, carrying the child's DNA, genetically distinct from the mother's own cells, do not die. They integrate into the mother's tissues, take up residence, and persist for decades. This is called Microchimerism, after the chimera of Greek mythology, a creature made of parts from different organisms. A woman who has carried a pregnancy may have cells from her child living in her liver, her lungs, her thyroid, her brain, her heart, detectable with sensitive genetic assays 20, 30, even 40 years after delivery. These are not debris. They are living, functional cells that have integrated into the mother's tissue architecture, dividing, producing proteins, participating in tissue maintenance. Some research suggests that fetal microchimeric cells migrate to sites of maternal tissue injury and contribute to repair, the child's cells healing the mother's body. decades after birth. If you are a woman who has carried children, cells from each of your children may be functioning in your organs right now. If you are a man, you are not a passive participant. Your cells crossed into your mother during her pregnancy with you and may persist in her body still. And the reverse is also true. Maternal cells cross into the fetus during pregnancy and can persist into adulthood. You, right now, reading this, may carry living cells from your mother in your own tissues. Cells that crossed the placenta before you were born, integrated into your developing organs, and have been functioning there ever since. You do not just carry your mother's genes, you may carry her cells, literally. In your liver, in your brain, alive. Let that settle. If your mother has died, some of her cells may still be living inside you, dividing, functioning, part of you in the most literal biological sense the word part has ever carried. Now consider your grandchildren. Each one received roughly 50% of their DNA from each parent, which means roughly 25% from each grandparent. But because of the same recombination that made you unique, the same chromosome-breaking, segment-swapping process that produced your 1 in 70 trillion genome, which 25% each grandchild carries, is unpredictable. If you have ever looked at a grandchild and seen your own eyes looking back, or noticed that one grandchild has your nose while another has your spouse's, you are seeing recombination's randomness made visible. One grandchild might carry the specific genetic variants that produce your eye color, your particular nose shape, your variant of a metabolic enzyme that determines how you process caffeine. Another grandchild from the same parents might carry none of those variants. A third might carry your risk profile for a particular condition, but not a single one of the facial variants that made your face look the way it does. You are not distributed evenly through your descendants. You are distributed randomly in fragments, a patch of chromosome 7 here, a segment of chromosome 14 there, in a pattern determined by the specific molecular breakpoints that form during meiosis in your child's reproductive cells. Each grandchild is a separate experiment using some of your source code, combined with someone else's, running in an environment you cannot control, producing a person who carries fragments of you in a combination that cannot be predicted and will not be repeated. When you see your own eyes in a grandchild's face, you are seeing a specific set of pigmentation genes that survive 2 rounds of recombination, one in your child's body, one in your grandchild's, to arrive at this particular pair of eyes in this particular face. The odds of that specific combination reaching this specific child were not 1 in 70 trillion. They were far smaller, because 2 shuffles separated you, not 1. What you recognize in that face is a fragment of your genome that made it through 2 molecular lotteries to be expressed in a person you can hold. Look at your hands again. What you see is not a copy of your parents. It is the output of a program that has been running for 6 decades, written in 3.2 billion base pairs, modified by methyl groups deposited by your parents' lived experience, shuffled by recombination into a combination that has existed exactly once in the history of the species, and now expressing delayed execution genes that were waiting since conception to reshape the tissue under your skin. The resemblance you see, the one that was not there at 30 and is unmistakable now, is not you becoming your parents. It is your genome revealing instructions. It has carried your entire life in its own time, on its own schedule, in a body that has never existed before and will never exist again. Those are your hands. They are also your mother's hands. They are also something neither of you planned and something no one will ever replicate. That is not sentiment. That is molecular biology.